So come to case 3. This case is actually a carrier screening. So we have a husband and wife. The husband had oligoasthenospermia, but was generally healthy. And in our carrier screening, we test for autosomal and X-linked recessive diseases with an early onset. And we look at 625 genes which are associated with this early onset. And just to give you a short introduction to the carrier screening function, and this filter is only applicable in case 2 individuals are related as partners and considered as unaffected. The filter will keep variants which are heterozygous within the same gene in both individuals. Hence, it considers variants which are potentially homozygous or compound heterozygous in the offspring. The filter also considers the PAR region and the X-linked diseases. So, of course, we start our case again looking for pathogenic variants, and this is again done by our customized filter button, and the inheritance filter here called the carrier screening. And what we found was, or was indicated to us, was a variant in the MUTYH gene, and this gene is associated with colorectal cancer, and its onset is already in the teens. So therefore, it's part of this carrier screening. And when you then when you, for example, analyze the mother here or the wife, you get an indication that the partner also carries this variant. In this case, 0/1 indicates heterozygous variant. And when you then look at the alignments, you can see that both wife and husband carry this variant. So in future offspring, there might be a potential pathogenic variant in the MUTYH gene, which might be then homozygous. But this was not the end of the case, because we also looked for high impact variants, which could be frame shifts, nonsense variant, or splice variants in the canonical splice site, for example, but also some others. And when we turn on this filter plus the carrier screening inheritance filter, we've found a variant in the col17A1 gene, and then when we looked at the alignments, we could see that only the wife carried actually this variant, while the husband didn't carry this variant. So we were actually a bit puzzled, because we didn't know why varvis was showing us this variant, and this would then be also my next question: what could be the other heterozygous variant in the col17A1 gene? And the answer would be the husband carries an EG missense variant, since it has a moderate impact. It's not shown by the high impact filter. The husband carries a CNV, or the husband carries an inversion. Okay, it's pretty much evenly divided. Only if you were going for the missense variant. So maybe you've seen it on the slide that it's indicated that the partner might carry a CNV, which is indicated here, and that was actually the case. When we then looked at the CNV module, we could see that there was actually a deletion affecting the exon 38 and exon 39. And again, this was shown very nicely using the neighbors feature. And when we then look at the neighbors feature, and when then look at the alignments, you could see here really nice the breakpoints within the alignment, and again affecting exon 38 and exon 39. So this shows that we had a SNV which affects the which is carried by the wife, and CMV carried by the husband, and together this potentially might be, these variants might be carried by the offspring in the compound heterozygous way. And this also shows that varvis can show you not only that two SNVs are carried by the wife and the husband, but also across those two models. So again, short summary: the carrier screening is enabling you to find variants which are potentially homozygous or compound heterozygous in future offsprings, and it's independent of SNVs or CVs. So just the overall summary: this inheritance filter, the inheritance filter, like the compound heterozygosity filter and carrier screening filter, and also the de novo filter or autosomal recessive filter, support you in your exome analysis. The HBO similarity score helps you to find the right genotype-phenotype correlation, and the CMV analysis works really well with the neighbors tool, and also so far we have found all the variants which were previously detected in a SNP array or AREY-CHH using the CMV model. So thank you very much for listening.